Today I'm going to be talking about some of the research that I did for my PhD, as well as some of the kind of background research that allowed me to be able to work on some really, really cool fossils. And I'll also be talking about a couple of things that are currently ongoing in my lab at Commonwealth University, Bloomsburg. So First of all, most important thing here, the world is colorful and animals help to bring a lot of those various colors out in the world, ranging from things like just these kind of dull browns that you see in female mallard ducks versus the iridescent speculum that they have on their wings and their heads, flamboyant cuttlefish with all their bright and beautiful colors, and all of the amazing colors that we see in geckos, various other lizards, amphibians, and various other invertebrates. And the world is just absolutely gorgeous, and we are lucky that we can actually see a lot of these colors that these animals produce. So there are two main ways that animals can actually produce colors. And one of those ways is via pigments. The other way is via structural colors. So pigments, basically how they work, they are chemical-based structures that absorb certain wavelengths of light and reflect other wavelengths. So for example, in this northern cardinal, the red feathers of that cardinal, we see that red because most other wavelengths of light are absorbed by the particular pigment that makes up those red feathers. So if the sun is producing white light, the sun is that white light has several different wavelengths of colors within it. Most of those wavelengths get absorbed, but the red gets reflected, and that's what actually meets our eyes and allows us to see that particular color. So our eyes perceive the red of the northern cardinal because it has a pigment in it that allows that red to be reflected back towards our eyes. As for structural colors, they work a little bit differently. You can have certain types of pigments in structurally colored feathers. We're going to focus mainly on feathers here. But the difference is, instead of just having pigments absorb certain wavelengths of light and reflect others, now we've got other structures that help to contribute to the color of those feathers. And some of those structures could be keratin, which is the hard part of the feather. They could also be this we've got this spongy layer here in a bluebird feather that's full of air spaces. So keratin, air spaces, plus certain types of pigments can cause a weird interaction of light that bends the light in such a way that we end up with other colors that aren't necessarily produced just by pigments. So blues, for example, most animals can't produce, or actually no animal, as far as I know, can produce blue pigments. Certain types of fungi can, but not animals. And so you end up with blues in blue jays or blues in bluebirds like this because of this structural interaction within their feathers between keratin, air spaces, and pigments. Same kind of idea for iridescent colors, only here we have layers of keratin, that hard structure that makes up the feathers, and melanin, a specific type of pigment that, depending on how you view the feathers, they could appear violet, they could appear blue, they could appear green. So iridescent colors change their color based off of your viewing angle or the angle of the bird's feather to how you perceive it. So a little bit different from pigmentary colors in that we've got several different things, several different layers that are contributing to the color rather than just one particular type of chemical. What I'm going to focus mainly today on are two different pigments called eumelanin and pheomelanin. Your hair, skin, eye color, weirdly enough, your liver color, all contribute or all have melanin in them. So if you have brown hair, for example, or if you have black hair, like my cat Adria here, she's a calico, she has black all along her arm here and along her nose. That is produced by a particular type of melanin called eumelanin. Her reddish brown colors, pretty rusty red orange colors that she has around her eyes and along her back here, those are produced by another type of melanin called pheomelanin. So if you have ginger colored hair or red colored hair, you have a lot of pheomelanin in your hair. So two different types of melanin that make up her beautiful coloration here. She also has white at the tips of her paws and next to her nose here. That is the lack of pigment. So there's no melanin in those particular hairs. There's no other pigments in those particular hairs either. So where you have white is where you were lacking pigment. So two main types of melanin here, eumelanin, which produces black and dark brown colors. It also very heavily contributes to the structural colors like blues and iridescent colors. And pheomelanin that produces rusty red and dull yellow type colors. Now, in most vertebrates, those two types of melanins are housed in organelles that we call melanosomes. Organelles just being tiny little subparts of cells that do a specific or have a specific purpose. So in this case, these types of organelles, they kind of look like this. These are housing those particular pigments. So they're housing the two different types of melanins. This is a scanning electron microscope image of a section from a mud puppy. That's what this animal is, where I took a look at the size and shape of the melanosomes that were found in mud puppy skin. So these are a nice little image of modern day mud puppy melanosomes. What's really cool is in birds, you can actually look at different colored bird feathers under a scanning electron microscope, a specialized microscope that shoots electrons down at your sample. And the melanosomes are different sizes and shapes. And those sizes and shapes correlate to the color of the feathers, which is really cool. So if you look at, for example, this is a tufted titmouse. We have little reddish brown orangish colored feathers here on the animal, and it has these little tiny meatball shaped melanosomes inside of those feathers. And yes, literally meatball shaped has been used in actual scientific papers to describe these. It's pretty amazing. But little tiny meatball shaped melanosomes, almost spherical in shape. If you look at the black feathers of, let's say, a cormorant like this, you actually have these more elongate pill-shaped melanosomes or pill-type melanosomes. That has also been used in actual scientific papers. So they're longer. This little characterization that I have over here kind of shows this. These are shorter. These are much longer in shape and more pill-like in shape. If you look at gray feathers, they have kind of a variation of this pill-shaped type where they're a little bit longer and fatter. And then really cool is iridescent feathers, like in this duck here, where you have very long melanosomes that are very thin in shape. And typically they are arranged in sheets or in crystal type structures like you can see here. So end to end, butted up against each other in a sheet-like orientation. So that's contributing to the iridescent color of those particular feathers. So in birds, again, the size and the shape of the melanosomes in their feathers correlates to the color of those feathers, which is pretty cool.
So there's a question in the chat.
Yeah.
White is the absence of pigment, but why is the fur not showing underlying colors in regards to the previous slide?
Why is the fur not showing underlying colors? Because her skin is kind of a peachish color underneath that, and the fur is thick enough that it's not going to show the peach color underneath. In polar bears, so polar bears we typically perceive as white, but if you actually look at their hairs individually, their hairs are almost kind of clear. And so they're just reflecting kind of this whitish color back at us, even though they're clear. So they're showing more of the underlying, whereas her fur is thick enough that it doesn't. I don't have a better way to explain that. It's the thickness of the keratin of the fur, if that makes any sense.
Oh, that's great. Thank you.
Good question. So size and shape of melanosomes correlates with color. What's really cool is hummingbirds. And I'll get to why I'm mentioning these guys here in a moment. But hummingbirds have these crazy shapes of melanosomes. So if you look at hummingbird feathers, they are highly iridescent. This one in particular is Anna's hummingbird, which lives out west, and I think southwest. But its feathers for the males, at one angle, the bird's feathers look pinkish or purplish, and at another angle, they're more of a dull green. So they have extraordinarily unique feathers. And part of that is thanks to the shape of the barbs of their feathers. So these little structures here are very flat on hummingbird feathers. And also they have a special shape of melanosome inside called, we call them platelet type melanosomes, where they're very flat, almost like orzo noodles, if you are familiar with orzo noodles. So very cool. And if you look at them under transmission electron microscopy, they're actually hollow, which also contributes to the color. So very highly iridescent animals, especially in the crown and throat feathers on these particular birds. So we'll come back to that here in a moment. So the whole point of this is to talk about filling in the fossil coloring book. And we're going to look mostly at dinosaurs with a couple of other extra things at the end. So since scientists first started studying dinosaurs in the early 1800s, we never really thought that we would be able to actually determine the color of any type of dinosaur. So a lot of people very early on, for example, we've got paintings here by Charles Knight and Rudy Zallinger, where they just depicted dinosaurs as these dull grays, dull greens, dull browns. And even in the 1990s, when you start getting Jurassic Park and Lost World Jurassic Park, they're typically shown as just these dull colors. So what we set out to do, and what others before me set out to do, was to try to determine, were they really these dull colors, or did they actually shine brightly like a lot of the animals that we see today? And so some of the early pioneering efforts into this, scanning electron microscopy was invented, I think, in the 1960s. It's a specialized microscopy technique where you shoot electrons at your sample and it shows you really, really tiny things on the surface of your sample. Like we're talking microns, nanometers, very, very small things that even the compound microscopes that you might be used to can't see. And what they noticed looking at fossilized feathers in 1995, using a scanning electron microscope, they noticed these little tiny microbodies on the feathers. And Davis and Briggs in this particular paper thought, okay, those might be remnants of decay bacteria. So after the animal died, its feathers got covered in bacteria and they started the decay process and then just got fossilized with the rest of the animal. Makes sense. Then in 2008, Jakob Vinther, who was actually Derek Briggs's PhD student at the time at Yale, took another look at these feathers, and he actually ended up reinterpreting those little tiny fossilized microbodies as melanosomes, those organelles that we have in our hair, in bird feathers, modern day bird feathers, that help to house the melanin that contributes to the color of those animals. And there's several different lines of evidence for why they are actually melanosomes and they are not bacteria. One of which that Jakob Vinther mentions in his paper is that if you look at this particular fossil feather from Germany, you'll notice that it has stripes. There's areas of the feather that are lightly colored and areas of the feather that are darkly colored. And what he noticed was that these fossilized microbodies only occur in the darkly colored areas. You don't see them in the lightly colored areas. Whereas if they were fossilized bacteria, you would expect to find them along the entire feather. And there's been several lines of evidence since then, including chemical evidence and some other physical evidence that unfortunately I don't have time to go into. But he reinterpreted them as melanosomes, and that opened up this whole amazing new field of paleocolor, which thankfully I got to be a part of for my PhD research. So his study led to an explosion in dinosaur color studies. And one of the first major dinosaur color studies was this one. This is Lee et al. 2010. And this dinosaur is called Anchiornis huxleyi. Anchiornis was a dinosaur that lived in China during, I think, the early Cretaceous, if I remember correctly. I might be wrong. But as you can see, here's the arms of the animal. Here's the torso and down into the legs here. The skull is separated here from the rest of the body, but you can see these gorgeous preserved wing feathers. They also had what we like to call these little feather trousers. So feathers coming off the legs. You can see some of the chest feathers here, crown feathers here around the skull. And they looked at these feathers under the scanning electron microscope and noticed, again, just like Jakob Winther noticed in his 2008 paper, these tiny little microbodies. And they actually compared them to, I can't remember exactly how many, but several modern day bird feathers. I think it was like 80 some modern day bird feathers, and found that they are similar in size and shape to the melanosomes in modern day bird feathers. And they actually did some statistical analyses to determine that this animal was colored something like this. So we had kind of mostly along the body, we had these fat pill-shaped melanosomes that correlate with the gray melanosomes that you find in modern day gray bird feathers. We've got these spots and spangles of black feathers along the wings and the cute little feather trousers that this animal had. And the crown had rusty red feathers along with some patches of rusty red feathers along the face as well. So this was the first dinosaur, or one of the first dinosaurs, I should say. There was a paper that came out 2 weeks before it that my advisor is still upset about. One of the first dinosaurs that was colored using fossil melanosomes and comparing them to modern day bird feather melanosomes. So where I should also mention, where they didn't find feather preservation, they assumed that those spots were white. So hence the white on the wings and the little feather trousers. This is another example. This is Microraptor, also from China, from Lee et al. 2012. And here's the skeleton of the animal. Tail is out this way towards the left side of the screen. Skull is over here towards the right side of the screen. And they looked at the feathers of this animal as well, and similarly found these elongate microbodies. Only this time, they're very, very long. They are arranged in kind of a sheet-like orientation. So butted up end to end against each other and next to each other, kind of like a sheet of paper. And so when they compared these particular melanosomes to modern day for bird feathers, they found that they were most similar to iridescent type melanosomes. And so they reconstructed this animal as having these beautiful iridescent feathers that would have changed color as the animal flew towards you, which is pretty cool. But wait, there's more. This is Kai Hong. This is one of the coolest color reconstructions that has been done so far. And it just looks like your normal everyday dinosaur. So Kai Hong, also a Chinese dinosaur. Here's the skull here, here's the ribs, one of the arms, the legs, and then the tail flares off back here with these beautiful palm frond type tail feathers. So gorgeous preservation on this animal, lots of really cool feathers. And so, of course, they did the same kind of analysis. They took tiny samples of these feathers and looked at them under a scanning electron microscope and found platelet type melanosomes, which you see in modern day hummingbirds. So this animal probably had melanosomes, probably had feathers that were very similar to modern day hummingbirds. And what's crazy is hummingbirds are the only animals that we see this type of melanosome in today. this animal, Kai Hong, wasn't that closely related to hummingbirds. So something funky is going on evolutionarily here. But this means that this animal had some gorgeous iridescence going on. And so they hypothesized that the animal might have looked like this, which is pretty amazing. Now, did it actually look like a rainbow? Probably not, but it had those highly iridescent feathers that modern day hummingbirds have, which is pretty cool. So those would have been found along the crown, upper wing of the animal, along its back a little bit, and then the rest of their samples were black. So pretty cool that you have an animal like this with those hummingbird type melanosomes. And I just saw something in the chat. Oh, wow. Yes, wow. Very cool. Why did the prior slide say non-avian dinosaurs? So this actually is a non-avian dinosaur. So all of the dinosaurs that I have mentioned so far are non-avian dinosaurs. That just means that they're not birds. So birds are the only dinosaurs that survived the extinction 66 million years ago. They are direct descendants of dinosaurs. So when we say non-avian dinosaurs, we mean dinosaurs that weren't birds back when they were alive, if that makes sense. So Kai Hong was not a bird, but it was really cool. Anyway, so we know that we find melanosomes in the fossil record. What about melanin? Can we find eumelanin in the fossil record? Can we find pheomelanin in the fossil record? And the answer is yes. So a number of techniques have been used to try to characterize melanins chemically in the fossil record. The most common of which is this crazy technique. This is called time of flight secondary ion mass spectrometry, also known as TOF-SIMS, which is way easier to say. And what this paper did, this is Lindgren et al. 2015. This is a bird, specifically a bird eye. And these are the melanosomes preserved in that bird eye. So they put this sample under the TOF-SIMS. I won't really go into how this works. Honestly, I'm terrible at TOF-SIMS. I don't understand it that well. But they compared the spectra that the TOF-SIMS readout gave them for the fossils, compared it to synthetic eumelanin and eumelanin from modern day animals, and found out that yes, eumelanin is preserved in this particular animal. The problem with TOF-SIMS is one, it's very difficult to interpret and understand if you don't know what you're doing. I have experience in that. And two, it's also very expensive oftentimes and difficult to use. So part of my PhD research was to set out and find a technique that's easier to use, easier to interpret, and doesn't cost nearly as much. So this is my baby. This is a bird that I worked on, 123 million year old bird from China. It unfortunately doesn't have a good name. We call it CUGB P1202 for China University of Geosciences, Beijing, P1202. Sorry, it doesn't have a nice name. But what's cool, here's the skull, here's its neck, the coracoids, and then here is one of the arms, here's the other arm, back legs coming down here. And there's two long tail feathers, here's the wings, claws, etc. So it's a very well-preserved bird. This is the, whoops, screwed up, sorry. This is the counterpart or the other side of the rock. So you can see the feathers over there as well. So very well-preserved bird. It's also a very small bird. And we actually figured out that it was a subadult or a juvenile, so not a full adult animal, which is interesting. And I'll go into why in a little bit. It also has these two elongate tail feathers, as I mentioned, which suggest it could have been male. Those might have been used as ornaments to attract females, as they are in, or as similar structures are in modern day birds. So we looked at its feathers under a scanning electron microscope, and we noticed these microbodies, which we interpreted as melanosomes. And what's cool is, kind of like Microraptor, most of the melanosomes that we saw were very long and arranged in a sheet-like arrangement. So we thought that the animal was iridescent in life, kind of like this. So kind of like a grackle or a European starling. Might not have been highly iridescent like Kai Hong, but still changed color depending on the angle it was viewed at. We also tried Raman spectroscopy on these particular feathers. And so Raman spectroscopy, what it does is very simply, it shoots a laser down at your sample, excites the bonds between the molecules on the surface of your sample, and gives you a readout that tells you what chemical you're looking at. So the nice thing about it is it's a lot cheaper than top sims usually. It's a lot easier to use and faster and easier to interpret. So what I did, these are modern day bird feathers up here. So we've got chicken, red-winged blackbird, house wren, Cooper's hawk, mallard, and wild turkey. And then these are the fossil samples down here. So 4 different fossil samples from different parts of the bird. And what you'll see is there's one peak here and one peak here. And those peaks match up to the peaks that we find in the fossil samples. So we do think that melanin specifically is preserved in CUGB P1202. So kind of a new avenue for people to look at pigments and characterize pigments chemically in the fossil record that's cheaper and easier to use than top sims. So very cool animal, 123 million years old. Absolutely loved working on this particular fossil. I will come back to its significance here in a moment. Basically, we know that melanin is preserved in the fossil record. We can find it with top sims, we can find it with Raman, we can find it with various other techniques as well. It makes sense because melanin, its structure is very complex. It is difficult to break down. It's a very large molecule. So not surprising that we have melanin in the fossil record. What about pheomelanin? Pheomelanin is still a little questionable. So in 2017, Caleb Brown et al. Found pigments related to pheomelanin, or sorry, chemicals related to pheomelanin in this cool dinosaur called Borealopelta. This guy is at the Royal Tyrrell Museum in Alberta. Highly recommend you go see it if you get a chance. But it's a little questionable whether or not Borealopelta actually had pheomelanin. In 2023, Tiffany Slater actually found biomarkers for pheomelanin. So again, chemicals related to pheomelanin in a bird called Confuciusornis, a dinosaur, a non-avian dinosaur called Sinornithosaurus, and an extinct frog. So possibly pheomelanin is preserved in the fossil record. It just still needs a lot of work done on it. Which leads me to, so we've got melanin, at least, possibly pheomelanin. We've got melanosomes. Why do we care? Like, what does this tell us about these extinct animals that we didn't know previously? So the big question that we'd ask here is, could extinct animals have used their colors for similar functions as modern day animals? And the answer, well, we have no reason to assume otherwise. So what does this mean for the various animals that I have mentioned so far? First of all, for Anchiornis has some weird color patterns. It's a very pretty dinosaur. So the hypothesis that Lee et al. 2010 posed for Anchiornis was that its spangles on its wings and on its cheek feathers here are kind of similar to what you see in nightjars. So nightjars are nocturnal animals. They use their colors as camouflage. It's very difficult to tell the difference between a nightjar and the trees on which it hides. So they hypothesized that Anchiornis may have used its colors for camouflage in a similar way. However, you've also got these kind of reddish brown crown feathers on this animal, which typically are associated with mating. So they can't really rule out that this animal might have used its pretty colors for mating as well. So we're not really sure there. Iridescence. So we talked about Kai Hong, we talked about Microraptor here. CUGB P1202 also. Iridescence in modern day birds is typically associated with mate choice. So the males tend to be iridescent. For example, European starlings, they're everywhere. They're bright, they're beautiful, they're unfortunately invasive. But the males tend to have that bright, beautiful iridescent, whereas the females tend to be more of a dull color. So it's mate choice, essentially. The more highly iridescent you are, the prettier you are to the females. So Kai Hong Microraptor CGB P1202 probably used their iridescence to attract females, which is cool. Kai Hong is actually the oldest example of iridescence that we have so far, and definitely the oldest example of those weird platelet type melanosomes. So that pushes that evolution back at least 161 million years, which is pretty significant. That's not exactly a young animal. CUGB P1202. Why this animal was so cool and so amazing to work on. This is my favorite fossil that I have ever worked on. So as I mentioned, we think that this was a juvenile or subadult animal. It wasn't quite an adult yet. We know that because it had pitted long bones. So what that means is as you grow, your long bones, like your femur, your tibia, your humerus, your radius and ulna, those fuse over time. And this animal wasn't completely fused. So its long bones weren't completely fused yet, suggesting it wasn't an adult yet. It was still growing. However, its feathers were iridescent. So that means that this animal was trying to attract mates, even though it wasn't a full adult yet. Modern day birds are the opposite. European starlings, grackles, all of those animals that are iridescent don't develop iridescent feathers until they are full grown adults. And then they start trying to attract mates. So this animal was opposite. So it was trying to attract mates when it was still young, a young type of animal. Opposite of birds, which is very cool and very significant, and adds to some really interesting studies of this particular type of bird that shows that they were very different from the birds that we have today, which is cool. Anyway, I loved working on this guy. So the next question that we need to ask, which dinosaurs can we color? Unfortunately, we can't color every single dinosaur. I wish we could. That would be amazing. First of all, most dinosaurs don't have preserved skin or feathers. So if we don't have this preserved skin or feathers, we just have the bone. Bone doesn't have melanosomes in it usually. And even if it did, that wouldn't tell us what color the animal was. Second of all, some of the dinosaurs that we have that did still have feathers preserved or feather-like structures preserved don't show off the beautiful array of different melanosome sizes and shapes that we have in modern day birds. They were all this kind of meatball shape, which could suggest that they were red-brown in color, but probably not. So there's a cool study on Sinosauropteryx, which is an older dinosaur that had feather-like structures on its tail and parts of the rest of its body, entirely covered in meatball-shaped melanosomes, might not have actually been red-brown in color. This is Bapausaurus. Honestly, I'm not sure if I'm pronouncing that correctly. Same kind of idea. And so what we have done, and part of my PhD research did this as well, is looked through the vertebrate evolutionary tree and determined which animals have melanosome variation that correlates with colors and which ones don't. And unfortunately, birds, closely related non-avian dinosaurs, so dinosaurs that aren't birds that are very closely related to birds, and mammals are the only ones that you actually find melanosome variation or size and shape variation that is also color correlated. There's some questionable stuff going on with pterosaurs, so I'm looking forward to seeing more of that. So if you think about pterodactyls, pteranodon, etc., cool research coming out about those guys right now that might also put them in the same category as birds and mammals. So cool stuff coming out. Unfortunately, we may never know what color certain types of dinosaurs were, even if they have feathers. I did see a couple things in the chat. One of the dinosaurs looked like an armadillo in shape. Do you think they were the same color as an armadillo now? We really couldn't tell. I'm not sure which dinosaur that is. That kind of looks like an armadillo. Like where is it? Borealopelta, this guy. Probably not. They're not very closely related to armadillos, but they could have been gray, or there are fairy armadillos that are kind of a pinkish color. It's possible, but unfortunately, we won't be able to know because they aren't very closely related to birds. So unless we find the pigments that actually gave those guys color, we probably won't be able to tell what color they actually were, if that makes any sense. Good question.
How do you take samples of such a fragile fossil for Raman spectroscopy?
Very carefully. We have these little tiny needles that we very carefully pry tiny, tiny samples off of the fossil feathers and put them on tape for scanning electron microscopy and for Raman spectroscopy. So very carefully, I just made my graduate student do that right before my daughter was born over the summer. So very, very carefully. It is nerve-wracking if you're not used to it. I was sweating like crazy. I was also pregnant, so that could have also contributed. Has anyone done this type of study on dinosaur specimens that have preserved skin? Skin. Dinosaurs, yes. The Borealopelta, that weird armadillo-looking dinosaur, that was its preserved skin. So yes, that has been done. I have also done some work on some amphibians, fossil amphibians and lampreys. So yes, one day those papers will come out. Is there evidence of sexual dimorphism in dinosaurs? Yes. So lots of hypotheses going on about sexual dimorphism. Sexual dimorphism, if you're not sure, that means that the males look different from the females. Specifically, there is a specific type of sexual dimorphism called sexual dichromatism. That's something that I really wanted to look into for my PhD, but didn't get the chance. So that means sexual dichromatism is where the males are differently colored from the females. And that's very possible. Nobody studied it yet. I wanted to do it in a type of bird called Confucius ornis, where we can tell the difference between males and females by looking at their feathers. But unfortunately, I didn't get the chance to do that. Hopefully someday someone will do that. But we will see. How much correlation is there between the pigments in different animal groups? Are the pigments closely related across phyla? It depends on the pigment. How much correlation is there between pigments in different animal groups? So mammals, insects, nearly every single type of animal has melanin, specifically eumelanin. A few of them also produce pheomelanin. And then melanins are also produced by certain types of plants and certain types of fungi as well. So melanin is quite ubiquitous. So it's very widespread across a lot of different types of organisms, not just animals. Other types of pigments, carotenoids, which produce beautiful red colors, yellow colors, orange colors. Those are also pretty widespread, not as widespread as melanins, but they're also pretty widespread. And then there's certain other types that are specific to, for example, parrots. Parrots, their red and yellow colors are produced by a type of pigment called psittacofulvins. Those are only in parrots. They don't occur in other types of animals. So melanins, carotenoids, and certain other types of pigments can be found pretty widespread. And then there are other types that are specific to certain animal groups or certain plant groups, if that makes sense. Did I answer everybody's questions? Does anybody else have any questions before I move on? Cool. I like the thumbs up. I do that too. Anyway, hopefully my answers made sense. All right, so unfortunately we can't color every single type of dinosaur and we can't color every single animal. One day that paper will come out. I'm working on it. Anyway, so switching gears to what I'm working on now, other than this paper. So I have moved on. I love birds. I wish I could work on birds. Unfortunately, as we all know, relationships with China aren't that great right now. So I have moved to invertebrates. I love invertebrates. They're really cool, and we can find colors of invertebrates in the fossil record as well. So dinosaurs and other vertebrates aren't the only animals that contain original pigments. We also have evidence in certain types of invertebrates. So for example, this is a really cool slab of an animal called a crinoid. They're also called sea lilies. They're still alive. They're just not doing as well as they did hundreds of millions of years ago. But this particular slab here, you can see we've got some whitish gray crinoids. There's some black crinoids. There's some darker gray crinoids. So same slab of rock, different colors. Doesn't make any sense unless you've got some sort of remnant of pigments left behind, which is really cool. So Chrissy O'Malley, who graduated from Ohio State right before I got there, did this study, and she found biomarkers for pigments, so chemicals related to these particular pigments in these crinoids, which is really cool. Nobody believed her. It took her forever to get this published after the dinosaur paper started coming out. This is another type of crinoid, similar age if I remember correctly, but this pink color that occurs on these crinoids, this is actually related to a particular type of pigment that these animals produce called quinones. So this pink color is caused by a pigment that has preserved for a few 100 million years, which is pretty cool. So there is evidence of pigments already in the invertebrate fossil record. What I'm doing is looking at some younger fossils and a wider array of fossils. So both of these studies are in crinoids. Most of the studies that have been done so far on invertebrate pigments in fossils have been done on crinoids. So I'm looking at some other things. When I was on vacation in North Carolina, in North Topsail Beach in 2018, I kept finding these cool guys on the beach. This is we call them echinoids. It's a sea biscuit, so related to sea urchins and sand dollars. It's about 30 million years old, and if you look in these kind of petal structures here, there's some purple. It's interesting. 30 million year old animal with purple in specific places on its exoskeleton. That's kind of cool. So turns out there was a offshore dredging project off of the coast of North Topsail Beach in 2015, and it released a whole bunch of these 30 million year old fossils onto the beach. And hundreds of them, so it released thousands onto the beach, hundreds of them have color patterns that are probably original to the animals, not just that particular type of sea biscuit, the Hemiptegus, but other types of sea biscuit and sea anemones, like Gagaria here. Sorry, not sea anemones. Oh my goodness. Sea urchins. This is a clam, an oyster shell from a type of oyster called a pycnodont. This is a sea star, specifically a brittle star that has pink pigments, sea stars with pink pigments. This is part of a crustacean here with spots, barnacles with beautiful stripes. All of these are 30 million years old, and they have yet to be studied. So I contacted Linda McCall, who is an amateur paleontologist in North Carolina, who has given talks on these before, but hasn't had the chance to actually study them. And she donated a whole bunch of cool fossils for me to work on. So that is what I'm currently working on. We're trying to figure out how these pigments preserved, if the pigments are actually preserved, and do some cool chemical work on them. What we think happened is that these animals were very rapidly buried in a storm. And so they were covered over, the rock cemented on them. And then 30 million years later, in 2015, they were rapidly dredged up by the offshore drilling project and rapidly put onto the beach. So you've got a mix of rapid burial plus not being exposed to the sun for very long or other elements for very long. We think that's why the pigments are preserved, assuming that they are. I still need to do chemical studies on them. But chances are, if you've got some really cool stripes and spots and stuff going on in these animals, chances are that's going to be original. We will see. There's an older site too, I should also mention. In 2022, I think it was, there was another dredging situation going on off of Holden Beach in North Carolina. And some of those fossils also have purple coloration to them, which is cool. These are 66 million years old, so they're twice as old as the North Topsail Beach material. So same age as the dinosaurs. I also have a grad student, Henry Davies. He's currently working on some fossil feathers from Colorado that Linda also donated. So these will be the first fossil color studies done in the United States, which is pretty cool. And then she also donated some insects that have some interesting stripes going on from the same site. So I don't have a student working on this yet. One day we will figure out what's going on with those insects as well. So ultimately, fossil color studies are important because they give us this really cool insight into the lives of animals that went extinct hundreds of millions of years ago that we never thought we could possibly understand. It tells us about interactions that they had with each other, members of the same species, members of different species, and how they might have interacted with their environment, their food, and otherwise. So glimpses into the past that we didn't think that we could ever have before, which is amazing. Also gives us a better idea of their anatomy, their physiology, and various other aspects of their behavior and evolution that we didn't think were possible before. It also, on the human side of things, helps us to understand how long pigments can last. So if you want to paint a bridge and you want that bridge's paint to last several years, add melanin. I don't know. But anyway, people always want to know the human thing, and I don't do the human thing well. So, cool stuff. I'm really excited about it. Hopefully you guys enjoyed it. Does anyone have any questions? I see. Let's see. Ekphora. Ooh. I can't open this picture. Do the snail shells have color patterns?
I don't know if they had color patterns, but they were a wide variety of different colors. I will send it to you.
That's cool. That might just be preservational unless they have like stripes or something. Oh, there it goes. Now it's opening. That's cool. Are those the Maryland state fossil?
Yeah, they're in the collection at the Calvert Marine Museum, and that is the Maryland State fossil.
That's awesome. Those are really cool. Anyway, I'll stop my share here. Any other questions? I see a hand from Jocelyn Pope.
Thank you, ladies and gentlemen, for having me up here. Yeah, patterns, about the patterns. Like their ancestors. So is it like showing like the patterns for the bones? Like the Mosasaurus.
Oh, I love your mosasaur.
Like so here's like part of the spine, like the neck and spine. So like here's the spine down here. Yeah, there's like a black line down here. Like black lines over crossing the line, like
Mhm.
arches as usual. So yeah, it's like the outside is like the skeleton. So it's like neck, ribs, tail.
Mm-hmm.
So it's like everything like here is connected. So the head, like the head, this, and the tail all connects. Plus, even about the amphibious ones. So it's like the skeleton. So here's like all the ridges. So like patterns. So
Mm-hmm.
so it's like brown, orange, brown, orange, brown, orange. So yeah, the Spinosaurus all show patterns. So yeah, technically movements are patterns because it's like, so like if it's like moving this way and it wants to eat, then it's gonna be like this. So left, right, left.
Research studies show the patterns on an animal?
So I know what you're thinking. Does the patterns show the study of an animal? Well, for starters, yes, it does, because patterns, as you know, I learned patterns in school though. But
Mm-hmm.
here's something
What's your question?
that
It's not showing.
is pretty much going to be curious. The T-Rex.
Oh,
So
that's
yeah,
cute.
technically, it's brown, just like Jurassic Park. But what's different is that it's like black here, just like the Mosasaurus. But it's not amphibious, just like the Spinosaurus, which is like right there. But it's gonna be like this. So patterns. 1, 2, 1, 2, 1, 2.
What's your question?
Mom, I'm giving a little presentation on
It's
my
not your
own.
presentation, baby. What's your question?
Do you mind? I'm the smart.
What's your question?
You are very smart. You were doing
So,
a fantastic job.
so yeah, feet are also a pattern. So it's gonna be like a checker placemat. So it's like boom, 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 boom. So it's like all gonna be like a beam, like on the ground. So it's like boom, boom,
Do you have a
boom.
question?
So yeah,
Thank you so much,
technically-
Jocelyn.
So yeah, technically that's all my little presentation. So yeah, bye ladies and gentlemen, I hope you have a great night. Thank you.
So I will say, Spinosaurus and Tyrannosaurus, unfortunately, we don't have melanosomes from them, so we can't actually determine what color they were. The Jurassic Park movies came up with those colors and patterns. Still plausible though. Mosasaurs, on the other hand, nice feather. Mosasaurs, on the other hand, some work has been done on their melanosomes. And you know how your mosasaur toy has a white underbelly? Its belly is white. That is real. So mosasaurs did actually have light colored underbellies.
Well, what do you know? History is finally made on these Jurassic Park toys.
Exactly. They listened. I'm so impressed. All right. Any other questions? I think I saw some pop up in the chat.
There's a few questions in the chat. Any speculation about how long ago pigments originated in creatures? What kind of age is the oldest pigment containing fossils we have?
The current oldest pigment containing fossil is the eye of a fish it's a little over 300 million years old. However, we do have, if you're familiar with the Burgess Shale, it's a really amazing site up in British Columbia in Canada. And it's got some of the oldest animals. They're about 508 million years old. They haven't found pigments in them, unfortunately. That's something that needs to be studied. But there are some cool microstructures that suggest that a couple of them were iridescent. So just a different type of iridescence than what we talked about with the feathers. So oldest pigments currently about 300 million years old. Oldest possible color about 508 million years old.
Wow, it's pretty
It's
amazing.
cool.
What about fluorescence that we can see in fossils? Is it just proteins or question mark?
There are several studies currently going on using, I forget what it's called. It's some sort of X-ray fluorescence technique. Michael Pittman is the paleontologist that uses it most commonly. But he has discovered some interesting, using his technique, he has discovered some interesting patterns in fossil skin. I can't remember if he's looked at feathers. In fossil skin and I think feathers that you can't see with the naked eye, that could be related to color. He has also discovered some soft tissues in fossils that you can't see with the naked eye using fluorescence techniques. So some really cool things coming out of there. The only problem with the technique that he uses is it sometimes burns off the surface layers. And so once you use it, you might not be able to use it again and get good studies after you use his fluorescence technique. But it's still really cool. I don't know if that actually answers your question or not, but it's cool.
Really fascinating to hear about. Do you have ideas of something accessible on this for a 10-year-old? Thanks.
Oh, let's see. There was, would that be accessible for a 10-year-old? There was a Scientific American article that came out on fossil color in 2018, I think it was. That's a little bit more accessible than an actual journal article. It's a really nice summary that came out in the magazine Scientific American, I want to say in 2018 or 2019, that summarizes the color studies really well. Otherwise, I wish I knew. I'm sure there are some really cool videos out there that EONS or other groups have done. I just haven't watched them. So I'm sorry, I don't know. I'm sure somebody's got some cool videos on fossil color. I just don't know who.
Is there any way to determine if a fossilized creature had bioluminescence that they may have used for communication of other purposes like flying squirrels, like modern marine species? Curious about crinoids, etc.
Not that I know of, but like I'm sure that extinct animals use bioluminescence just like modern day animals do. But as far as I know, there's no actual evidence for that. We can just guess based off of evolutionary relationships. That would be really cool to find out. But I don't know how you'd go about doing that. Probably some sort of chemical study. I just don't know how.
Earlier, you showed structure inside bird feathers that cause refraction of light, causing iridescence. Have you seen similar structures in fossil feathers?
We have seen the sheets of melanosomes that you typically see in those iridescent feathers. Keratin, so the hard part of the feathers is a little bit questionable as to whether it's preserved or not. So keratin might be preserved, it might not be preserved. That's a huge argument. But we don't see the layers of keratin versus melanosomes that you typically see in iridescent feathers. We just see the elongate sheet-like melanosomes, and we see in Kaihong the weird hummingbird type melanosomes. But like I said, not keratin, and if we don't have the keratin, we can't see air spaces like you would see in non-iridescent structurally colored feathers either.
It looks like one of our club members might want to collaborate with you on working on something to translate this information for the younger audience.
That would be awesome.
Oh, and someone found the Scientific American article and posted it in the chat for those who are curious. I'll also share that information in an email to everyone who's here.
Oh yeah. I should add eggshells to this talk because that is really cool. That's a study by Yasmina Wyman, who's currently at Johns Hopkins University. She's got some cool stuff on fossil eggshells with preserved pigments in them. So yeah, it's very cool. Very cool stuff. Yeah, any other questions? Thank you.
Are there any other pigments that might show up in fossils or?
That is something that I really want to look at. So melanins, definitely, at least eumelanin, possibly pheomelanin in vertebrates. Invertebrates, right now we have quinones, which are making the pink colors in the crinoids. I suspect that we will find other types of quinones in the fossils that we're currently looking at. I'm also guessing we might find some carotenoids, which are the second most common type of pigment in some of those fossils as well. So that's something that I'm hoping to determine in the relatively near future. Are there other pigments preserved other than just quinones and melanins? And then also the biliverdins that Yasmina found in her eggs as well. So hopefully, cross your fingers.
Excited to find out. As far as the birds go, I thought it was really interesting that the specific one you were working on, you could tell that it was a juvenile and the colors were specific to that. Are there any other fossilized versions of that species that might have different color patterns that you could compare it to?
I wish. That's why he doesn't have a nice name. So it's CUGB P1202. That's the designation in the museum that it's housed in over in China. Since that particular type of birds called the bohyrhnithids, that particular type of birds is completely extinct. There aren't very many fossils of those particular birds. Currently, I want to say, I could be totally wrong about this. I want to say that there are 7 species that have been described, probably more now. And this was the youngest at the time, the only one that wasn't a full adult. So we couldn't determine if it belonged to a species that already had been described or if it belonged to a new species. Hence, it doesn't have a nice, pretty name. None of the other bohyrhnithids, unless some of the most recent ones have had this done yet. I don't know if they have or not. But none of the other bohyrhnithids, as far as I know, have had color studies done on them. Quite a few of them don't preserve feathers. There are a couple that do. So maybe someday someone will compare CUGB P1202 to those. But as far as I know, nobody's done that yet. We will see.
It would be pretty amazing if you could find like a male and a female of a specific species and then be able to see if there were color variations like there are with modern day birds.
So there are thousands of specimens of a bird from China called Confuciusornis. And you can tell the difference between male and female Confuciusornis. The males have the long tails, the females don't. And there are a couple of specimens of females that also have eggs still inside the bodies. So you can tell that they were female. It's just nobody, when I asked my advisor during my PhD if I could study the Confuciusornis, he said somebody else over in China already was. If he was, that paper hasn't come out. So that would be a really amazing study to look at the color patterns of the females versus the males in Confuciusornis, but nobody's done it yet. Sadly.
Well, I hope someone does and publishes it because that sounds really fascinating.
Yeah, we did look at one specimen of Confuciusornis. We published it in 2018, but we didn't compare it to other specimens. So there you go. Any other questions? Cool. Well, thank you so much for having me.
Thank you so much. This was amazing. Oh, we have another question.
How did melanosomes survive the fossilization process? So that is another thing that we are currently trying to figure out. Some studies have been done where I didn't do this, I shouldn't say we, but they placed feathers, modern day feathers in specialized autoclaves. Autoclaves are used normally to subject things to high pressures and high temperatures to kill off bacteria. So they placed the feathers in specialized autoclaves with specific pressure temperature settings to mimic fossilization process, and just found that the melanosomes survived. It's probably because the melanin inside is such a complex chemical that it's very, very difficult to break down. I will also mention that was eumelanin that survived. The pheomelanin didn't do so well. So eumelanin just has that structure that allows it to be preserved, whereas pheomelanin maybe does, maybe doesn't. So that complex chemical structure is what allows the melanin and melanosomes to survive, which is cool, as far as we know. Any other questions?
That was an awesome presentation. Thank you. Really,
Thank
really
you.
good.
I love talking about this stuff. It just makes me so happy.
Your enthusiasm is really infectious.
Thank you. Thanks for inviting me.
Well, if anybody thinks of any questions later, feel free to email them to me. I can get them to Jennifer or pass you her information. And I look forward to your research on the shells that you're doing. It will be very exciting to find out what you discover.
Thanks. I'm excited. I can't wait to get going on it.
Well, I hope you have a good evening. Thank you everyone for joining us, and keep your eyes peeled for emails about future upcoming Fossil Club events.